You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast. I have Dr. Abby Johnson. She's a postdoctoral associate in the Knight Lab, the University of Minnesota. The Knight's Lab, I'm sorry, not Knight Lab, but Knight's plural. She's studying the relationship between diet and the microbiome. Thank God someone's doing this, because a lot of people say, "Oh, diet doesn't have much effect," which I think is absurd. She received her Ph.D. in nutrition from University of Minnesota, where she studied the impact of bariatric surgery on body composition. Very cool. She's a registered nutritionist. A lot of great background, a lot of great credentials. So, Abby, thanks for coming.
I'm happy to be here.
Yeah. So, tell me what spurred your interest in the relationship between diet and microbiome?
I've been really interested in the diet and the microbiome probably since about 2010, 2011. I spent a year at an institution at the University of Chicago, and while I was there, there were some people who were studying the way different fats were impacting specific bacteria in the gut and potentially relating to the development of different bowel disorders. And it blew my mind that the food we were eating was changing the bacteria in our guts. And this was right around the time that the field of the microbiome was really ramping up. And that was what triggered my interest in microbiome and diet. I had already been interested in diet, but the microbiome seemed like this clear link that we'd been overlooking for so long that could explain a lot of what we didn't understand about diet and like different diseases, the way people develop obesity, all of that. Those sorts of pieces seem to make sense to me that, there are other things living in us that probably matter. So,
it's funny, like my wife has made kombucha and, we saw the SCOBY and it kind of freaked her out, the bacterial colony that lives in it. And, I learned about fermented foods and I realized like literally everything we eat has been worked on by bacteria or fungi or yeast. And even once we eat it, it's still worked on by that stuff. There's literally nothing we eat really if it starts out antiseptic, it doesn't stay that way for long. So the microbiome is like huge.
Go on, sorry.
I was saying the role of it is it's like, it's so intertwined with everything. It's just, they're a part of us. They're a part of our metabolism. They just have a tremendous role. So I'm sure diet is everything for them.
Yeah, and I think there's some really cool research that's coming out that suggests that lots of the food we eat, even the things that aren't fermented, also contain microbes. So this idea that anything we eat is actually sterile is, under threat right now. So I'm really excited to see where that goes and how that changes our understanding of diet and the microbiome in the future.
So what's some of your specific research about? What are you looking into or hoping to figure out?
Yeah, so my research is really centered around this question, what should you eat? And that question obviously varies depending on what you're interested in, right? What should you eat for long life? What should you eat for weight loss? What should you eat for gut health? Everybody has their own personal ending to that question. And as a dietitian, I'm really interested in being able to translate research that we're doing in the microbiome to people to actually try to help answer that question. Because right now, if you ask somebody, or if somebody asks me, what should I eat for my microbiome? I have a hard time answering that question, even though I spend all of my time thinking about the diet and the microbiome. There's not really strong, solid data for any one thing being beneficial for everything. So I'm interested in developing, primarily my research is to develop methods that will help us take the research that we're doing to that step of being able to translate it to people. So we can say, oh, you are trying to change this part of your microbiome. Here are actual foods that contain the compounds that your microbiome needs, and these are the things you should be eating. So I develop computational methods that let us assess your diet differently or in different ways than we have been doing to take into account the foods that you eat, so that hopefully on the far end in the future, when we're doing more research, we'll get to the point where we can say apples are good for you and pears are good for you and broccoli is good for somebody else. So that's really what my research is aiming for. And most recently, I've been doing work on a longitudinal cohort, but it was a short time longitudinal. We collected very dense data for two and a half weeks on 34 people. And a lot of my work at the moment focuses on untangling the networks of diet and microbiome that we have for these people.
Well, when someone says what food is good for me, I would think the person obviously is thinking of themselves and not their microbiome. And even answering that question, what's good for you? I mean, okay, is it true that what's good for you is always good for your microbiome, or can the two be at odds? Like, how can you reframe the question where you can get better results than the way everyone else asks it?
I don't know. I'm not sure if what's good for you is always good for your microbiome or vice versa. And I think that's a really good question and one that we should be asking. What's challenging is that, and I'm sure you've heard this from other microbiome researchers who you've talked with, is that we don't know what good microbiome is. We're still trying to figure out what is healthy when it comes to the microbiome. So a lot of it depends on that being worked out first.
Yeah, all I hear is, you need a diverse microbiome. Diversity is good. I don't know what that means. Diversity in terms of just more species being there, or diversity in terms of like the redundancy of certain species or strains to produce certain metabolites. I don't know. I mean, people, again, throw that term around, but I don't know what it means.
Yeah, I think when people talk about diversity, both of those features that you mentioned there, numbers of different types of species, and also the different types of things that those species can produce as a community, both of those things are considered in terms of diversity. The other thing that we've been exploring in the work that I do is a question about what about stability? Does your ability for your microbiome to rebound after a perturbation or an assault, does that mean something for health? And that's something that we're actively investigating as well. Can we change the stability or your ability of your microbiome to bounce around from day to day, from one state to another, and to recover from an assault like antibiotics? Does that resilience or stability matter?
Well, it seems like, I don't know if this is right, but it seems like, the microbiome, like, let's say in our gut, it's like a job center, and our body needs certain metabolites and, functions fulfilled. And in return, instead of the bacteria being paid, they're being paid with, food and a place to hang out and live in peace. So I guess one reason maybe for the redundancy is that not just one microbe can make, this short chain fatty acid, but there's a bunch that can. So a community gets set up, they're trading resources, they're doing their thing, they're helping us, we're helping them. Then maybe you take an antibiotic, it disrupts the community, and other actors come in to fill those necessary roles. They may be different actors, and these actors may be like real curmudgeons and not be cooperative like the other actors were, or maybe they can't produce certain metabolites that they also were producing that we needed. So maybe that's where the disruption of the community comes from. I guess what I'm saying in summary is, maybe if we look at it as like in terms of the metabolites and the function of the whole community as a whole, instead of like the actors in it only, maybe that gives us a different picture of what goes on.
Yeah, I think that that's absolutely worthwhile. And I think that's the direction that a lot of us are heading in now, is to move beyond just what was there in your community, but what were they doing? And it's a much harder question given our current technologies. And I think that's why for so long now people have been focusing just on the who, but the what and the what are they doing? Those questions really matter as well. I'd like to add a little bit of complexity there too, in that when I think about diet, we have been reducing diet to a series of macro and micronutrients, the things you're used to reading on a food label, the things that current dietary software will spit out as this is what your food contains. So if you think about a strawberry, you'll probably be able to think like, oh, okay, a half a cup of strawberries, maybe 50 calories. Most of it's from carbohydrate. There's fructose and minimal amino acids. But if you're a bacteria and you're living in a gut and you see a strawberry come by you, that doesn't look just like our nutrition label. That strawberry contains many different phytochemicals. It has slight variation in the fiber compositions. So maybe instead of a chain of carbohydrates going off to the right, they go off to the left, and that's specific to strawberries of a specific strain. And maybe bacteria care about that type of complexity. So when you're talking about wanting to understand the complexity of what do bacteria do, not just who's there, I think we also need to consider with the food that we eat, what really is the food that we eat? And when the bacteria sees it, how is it different? Because if in the upper gastrointestinal tract, a lot of the things that we as human hosts need has already been extracted from that food, what remains when the bacteria sees it is...
Well, I mean, there's bacteria the whole way. In our mouth is ones that are getting first dibs on it a little bit. And then, down our throat, our, esophagus, there's ones there. And then, in our stomach, there's other ones there. And then in our...
Right. There are bacteria the whole way through, but the majority of the bacteria that live inside us, like when I think about bacteria in the gastrointestinal system, like the vast majority are in the large intestine. So that's what I was talking about. But yeah, you're right. No, there are bacteria the whole way down the line. Some are maybe more important than others. Some are doing things that we don't yet understand because they're harder to reach and harder to sample. So yeah.
I just had a strange thought. Fermented foods seem to be, really good for us. But, is it because the bacteria, for instance, in, I don't know, sauerkraut are producing beneficial compounds for us? Or is it the bacteria themselves that are being used as a food source by us and by other bacteria? Or is it that the bacteria in, let's say, a sauerkraut survive their way into our body and help populate our body more with that particular bacteria? It seems like there's multiple ways to look at it.
Yeah, and that sounds like an excellent study, right? Where we should feed people like cabbage and sauerkraut and see what's different. How does it change the microbiome? Does it improve or change a health metric? So I don't think we've seen the studies that we need yet to answer that question of like, why are fermented foods presumed to be good for us in a health beneficial, right?
Yeah, it's weird.
Yeah, and nobody's done the research. So we've got like huge opportunity to work these things out. And I'm not sure that we have really solid research yet to say like fermented foods are always good either, but I think we have good basis in evidence from other things, from previous epidemiological studies that suggest that yes, fermented, but I don't think we've studied them closely in relation to the microbiome. We're close enough yet in relation to them. I think people are doing those research studies now.
Yeah. What about people that eat a very narrow diet? They only eat a few things versus people that eat all kinds of stuff. What do their microbiomes look like, and why do you think?
Yeah, so I can speak to one specific example from my recent research. So in my most recent research study, we recruited 34 people to take part in this study, and they collected a fecal sample every day for 2.5 weeks. And we were looking at this outcome of stability. How stable was your microbiome? Did your microbiome move around a lot from day to day, or was it pretty much the same every day? And we, in our study recruitment, we ended up with 2 people who, I work in kind of a computer science field, and 2 of the people who were in my study, turns out they only drank this meal replacement beverage called Soylent. I don't know if some of your listeners have heard of it, but I've tried it, and it's an interesting flavor profile. But it's just this meal replacement beverage, right? And so 2 of my participants, that's all they consumed for the entirety of the study. And when I got this data back, what on earth is going on? Like, why do their diets look so weird? And when I figured it out, I just kind of had this, oh man, like what were you thinking? But we had this cool little subset, right, where we could look at these two people in relation to the rest of our study and find out whether or not their microbiomes were, say, less or more stable. And what we found that I thought was interesting and needs to be tested in a bigger study is that their microbiomes just looked like everybody else. They weren't different in any way. Their microbiomes were right bang smack in the middle of the stability variation that we had in our study. And so I'm not sure, because it wasn't a study about drinking Soylent, right? It wasn't a study specifically designed to see what happens when you only eat one thing. But we had this signal in our data that suggested that people in our study who ate many more different things each day had more stable microbiomes, but the reverse wasn't true. The two people we had in our study who only ate one thing, their microbiomes were not different. They were just as everybody else. So we've got more to figure out with that question. But I suspect that meal replacement beverages actually contain enough different types of substrates for bacteria that they were able to somewhat maintain the normal sort of status quo. I'd be really interested to know what happens, though, to people in a hospitalized setting. A lot of people in hospital who have various nutritional conditions or conditions that affect their nutritional status end up on essentially meal replacement beverages, right? They take enteral nutrition formulas instead of eating. And it'd be quite interesting to look and see how those microbiomes or how that affects the microbiome in people who aren't healthy and people who have other things going on, whether or not.
I was going to say, hospital food's so bad, the bacteria probably commit suicide and leave.
Yeah, and this is like, what is extreme? This is the extreme end of hospital food, right? Like this is hospital food in a tube. So it'd be a really cool thing to look at to find out what happens to the bacteria of people who are already under stress and then are either eating hospital food or are eating meal replacement beverages. So they're either getting diversity or they're not.
So going back to your study, so the people that ate only Soylent had their bacterial profile looked healthy, is what you're saying?
Yeah, it just looked like everybody else. And I'm not going to make a claim that Soylent supports a healthy microbiome, but
unless you work for them, yeah.
I don't. Full disclosure, do not work for Soylent, but I think it's interesting that their microbiomes didn't crash during the study. I suspect they were people who consumed Soylent regularly, but I suspect that they took it to the extreme. My study knew that they were being studied, so I don't know how that impacted things. Like if it was a person who'd never consumed Soylent before, and I'm not sure.
Well, okay, so what was the difference you saw? So Soylent didn't affect it. Soylent only. What did affect what?
Yeah, and it's a funny study because it was observational, right? We let people eat their usual diet, and we just asked people for a daily diet, a daily microbiome, and tell us everything that they ate for 2.5 weeks. The big finding that we had in our study was that if we knew what you ate in terms of the foods that you ate, then we could pair your microbiome with your diet. Like we could connect whose microbiome samples were belonging to the person who had eaten what things, but only if we knew the foods that you ate. If we tried to connect your microbiome with your nutrient content, we couldn't match you up. So what mattered for your microbiome's composition in our study, what we figured out when we looked longitudinally in the data was that you had told us that we knew what foods you ate. And if we knew the foods you ate, we could do a better job than if we just knew your microbiome in predicting how your microbiome would change the next day. So we were able to improve this ability to connect diet and microbiome and identify how the foods, or identify that there was a pattern in the foods that were being consumed and how they were causing fluctuations in the microbiome over time. And just
what's an example?
So because it's kind of abstract, it's hard to give a specific example. But we found that everything was very personalized. So it mattered what your microbiome was and what your foods were. We tried to connect foods with specific species, and I'd hoped that we'd have enough overlap in foods that people ate. Naively, I thought that people ate similar things, but it turns out with 9,000 food choices in a supermarket, everybody eats a totally different diet, and there's very little overlap, even from between people who live in the same household. So what we did is we sort of went up a level of the food. So instead of looking at foods and specific foods, we kind of looked at foods in terms of very specific food groupings. And we asked whether or not foods from the similar food groups, if they resulted in the same changes in species compositional or species amounts in people, and if those things were true across different. And in some cases, we found that, for example, specific type of Roseburia species seemed to increase in everybody who had it, who also ate cakes and cookies and grain sort of bars, like meal replacement bars. And so we thought that was interesting. But in lots of people, the same food resulted in totally different directions. So I think we had dark green leafy vegetables in one person would mean that the same species of bacteria either went up, or in another person, dark green leafy vegetables result in that same species of bacteria going down. So what we identified was that responses to foods were personalized.
So that you couldn't find any correlation with some things?
Very few correlations that were repeated across people. I think that this would be maybe slightly different if we had controlled diet perfectly. We'd have more power to detect these differences. But given the data set that we had, we were only able to detect certain differences, and the differences that we could detect within a person, they were variable across people. So it seemed that your personal microbiome, that community that you have that's very unique from other people's, that those communities responded differently to the same foods.
Yeah, I mean, you could reslice the data a lot of ways. Like if you went back and, I mean, how many ways did you like slice and dice this data? Like what if you looked at macro ratios, calories, timing of the eating, man or woman, age?
We haven't done everything, right? Like as a scientist, you want to be careful not to just like search and search and search and search until you find something. So we made hypotheses and then tested them, but the data is publicly available. And if people are interested, they're like, and they have the skills and they want to like dig into it, it's all out there. And I'm happy to share it with anybody who wants to start like asking some of those more specific questions about like, oh, did it matter if you had various like macronutrient ratios of carbohydrates to proteins, or was the timing of the meals that you consumed important? And all of those things are like, it's in the data. It's a really nice data set.
Well, in general, do you see a lot of variation? Like, okay, well, two questions. Did the people consistently eat what they were eating for the whole 2.5 weeks, or did some like radically change?
People's diets changed way more than you'd ever imagine. People eat.
Oh, really?
Yeah, people eat, like the people in my study, their food intake was highly variable. So I think that...
Except for the Soylent people.
Except for the Soylent people, yeah. There were some people who were less diverse than others, but for most people, it was highly variable from day to day. Much more variable than the microbes. So the microbes in most people's guts stay relatively consistent over time. But the food that we eat, and you can kind of think through this probably in your own life, like if you go out for dinner one night with friends versus when you're home eating by yourself, just what you ate yesterday is probably not the same as what you ate today, unless you're a bodybuilder who's training for something. Your diet, we seek out variety. So it was much more variable than I ever anticipated.
So what kind of variability did you see in the people's microbiomes?
I'm not sure I understand the question.
Oh, so were the same species present the whole time? Was it just the percentages of each species? Like what did change?
Yeah, so for most people, the species membership stays relatively consistent throughout the 2.5 week period in our study. It's just like minor fluctuations. Some things go up, some things go down. And that's what seems to respond to the foods that you...
Okay, so it was just a fluctuation in the percentage composition of who was there. But who was there stayed the same.
And I haven't like dug deeply into the data to see if there were, within a person did they have a little bloom of something that not being there and it just showed up and then it disappeared. I think like looking for those types of things in the data would be really interesting, but it wasn't part of what we did initially.
Yeah.
Yeah.
But in general, if you were to sum it all up, not much changed.
Yeah, not much changed. Even though everything that people eat is wildly different.
Huh.
Yeah.
What about the, you said what they ate changed a lot, but did it follow any pattern, or it was like, I mean, I would think that it would be variable, but it wouldn't be all over the place. Like someone that's, a vegetarian certainly wouldn't all of a sudden start eating a lot of meat. So I would think like people have variation, but variation within some kind of like set band of eating.
I think that's really accurate. I think that there are two things at play when we think about diet and the microbiome. And one is like this habitual, like you just identified, like this band of what you normally eat, of like what is normal for you, that you might capture if you ask like really broad questions like, oh, do you like sort of rice and pasta and starch? Or are you like, do you trend towards only eating vegetables, or do you avoid meat? Like some of these big sort of overriding preferences that we have. But then there's this day-to-day fluctuation. And the study that we did was more looking at that like tight day-to-day variation. But you're right that people have this band of like probably normal, and the shifts in those types of eating patterns take longer, right? So it could be that those types of eating patterns, like your general normal eating patterns over time, might be more important for shaping your gut microbiome. Even though the small day-to-day variations, we were able to find that those also changed the microbiome as well. So you can imagine you can like link these two things together to actually come up with diets that could make small incremental changes to shift communities like in a direction that you wanted, if you knew what direction you wanted to go in.
Well, also, did you do 16S sequence or shotgun?
We did shotgun. So we did shotgun.
Okay.
Yeah. So we had about 580 microbiome samples, and they were all sequenced using shotgun sequencing. But we did this more experimental or sort of relatively newer version of shotgun sequencing, which we call shallow shotgun sequencing. So we didn't go as deep as many people often do. The lab that I work in is really focused on perfecting the technique of determining who's in the microbiome from fewer and fewer actual reads, so that we can get down to a nice specificity without having to sequence really, really deep. And that saves us a lot of money and allows us to do studies like this that have dense longitudinal sampling for around the same cost as it would take for 16S.
Yeah, because I was going to say that would have been a lot of money.
Yeah. If we had gone like deep, like couple million reads per sample, it'd be quite expensive, at least with the technology that we had available at the time when we did the sequencing. I think now with our most recent study, we collected like 9 samples per person. I think we got about 6 million reads per sample. So we're getting good at now getting like pretty good depth. I think this study that I've been talking about, we got about 500,000 reads per sample. So it was really shallow.
What about metabolomics? Did you look at that, or the gene expression? Or
no? We have duplicate fecal samples for each sample in the study. We could look at metabolomics, but we haven't.
Yeah, I mean, so right, you did this study. A lot of effort went into it. So, what does it tell you now to look at, or are you going to move on to like other areas? And where do you think you should return to it, maybe, and do metabolomics, look at gene expression? I don't know, other elements of it.
Yeah, I think. What I'd like to do next is something more well-controlled to like figure out ways or when we put people on controlled diets and do interventions that can, if everybody gets sauerkraut and I'm controlling everything else that they're eating, can we pick up a signal for sauerkraut? That would be, I think, really beneficial and help move the field forward. Those are the things that I think we need to do next. And that's where I'd like to spend my efforts. I don't know that I want to go back and sort of use this biobank of samples that I have to explore things. I think that would be a bit sort of fishing-y since I don't have a strong hypothesis. Yeah, that's what I was going to ask you. So you're not sure, like, I guess this didn't give you enough of a signal to say, now what do I do? Yeah, I think the only thing that we picked up that was really interesting signal-wise that it would be worth exploring in the future is this question about stability. I'd really like to look into this dietary diversity stability question more. Nobody's been measuring stability just because most people have been limited to sampling at one time point, right? But if you can get a kind of metric for how much a microbiome is changing from day to day, then you could start to ask questions or try to shift that to make somebody's microbiome more stable. Does that improve health or does it worsen disease, right? So those are questions I'd like to explore.
Yeah, there is a show, Bizarre Foods with Andrew Zimmern. Mm-hmm. And he goes all around the world. And, this is how nerdy I am. I'm watching the show with my wife and I'm like, wow, he must have like the most diverse microbiome in the world. He goes all over the world eating food. Yeah. So his poor microbes, Who knows what they're getting, I mean, everything.
But maybe that's good for maybe that means that he's got lots of competitive relationships between the bacteria that live in his gut who are fighting over these weird things that come down, and they're his microbiome is unusually happy. I don't know.
Yeah.
But
I just think he'd be a really interesting person to sample because, I mean, he's had everything in his mouth. He's eaten all kinds of foods. So.
I think he's in Minnesota, so maybe I should try to recruit him.
It'll be funny to have him as part of the study.
Yeah.
He'd certainly be willing to eat what you tell him to eat.
Right.
He's eaten all kinds of things.
Right. Michael Pollan did, right? He's got his microbiome as part of the American Gut Project. So maybe
really?
should be next. Yeah.
Yeah, that's cool. Huh. So where do you go from here? You said that you want to do a study to look at, again, stability and under what situations stability falls apart, I guess. But is that encapsulating where you're going to be taking things over the next year or two?
Yeah, I'm not really sure. I'm a postdoc in my career, so my next step is to figure out where I go after. But I'm kind of toying with of exploring this idea of microbiome stability and disease states. I'm interested in whether or not it's affecting diabetes. I'd like to tie it in understanding how it impacts diet microimmune relationships and what those are affecting frailty and aging, things that I'm a lot at, but there might be signals that suggest that there are relationships. So those are questions Do
you see any area that no one seems to be paying attention to that you think is critical to study? I know you can't do everything.
Yeah. People ignore. I think we need to I know a couple people are starting to, but I think run every mass spec and get big food data. We have the food omics to go along with our other multi-omics. If we're going to try understand stuff and use machine learning pipelines to get robots to do everything for us, then I think we need to know what's in our food beyond just carbohydrate, fat, protein.
Right. Yeah. It's nothing.
Yeah.
Huh. Okay. Well, very good. What's the best way for people to get in touch and to maybe give you unasked for advice on what you should do, or find out about what the experiment you've done?
I think it's probably best if people who want to reach out can probably reach out to me over Twitter. I'm always checking Twitter. I'm not super active in posting Twitter, but I believe I have a Twitter handle. I don't want to give out my email address, but I think it's Abby Johnson RD. Let me confirm. Okay. That's actually my Twitter. You can edit this.
Yes. So for the editor, make sure you edit this out and make it clean. It's okay.
Got to Twitter and realized I don't know where it says what. Yeah, so my Twitter handle is at Abby Johnson RD.
Okay, very good. Abby, thanks for coming. It's been a really great call. I appreciate you being here.
Yeah, thank you very much for having me. These are really wonderful questions.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.